So we are very lucky to have Dr. Richard Mooney here with us today for Frontiers. Rich is the George Barth Geller Professor of Neurobiology and Director of Graduate Studies at the Duke University Department of Neurobiology. From a very young age, Rich has been fascinated by two things, which is natural sciences and music. He pursued both of these as an undergraduate at Yale University, where he studied classical guitar, as well as studying how insects, auditory processing of insects. Following Yale, he moved, in addition to a couple of research gigs, to the San Francisco Conservatory of Music, where he studied classical guitar. And in 1984, in a state of artistic satisfaction and extreme financial hardship, he approached Jim Hudspeth in the UCSF Department of Physiology, working in his lab, working on auditory processing as well. That led to him doing his doctoral work at Caltech with Dr. Mark Kanishi, where he first began his work on the songbird, which we're very lucky to be hearing about today. Following a postdoc with Carlos Schatz at Berkeley, he moved to Duke University in 1994, where he's been ever since. His research, it's amazing and it's a real inspiration to me. One of the things that I really love about it is Rich's consistent ability to connect really fine micro-level investigations of neural circuits to the sort of memory and learning capacities of the brain that really motivate a lot of us to study neuroscience. In addition to his research we're about to hear about and the accumulation of awards that are way too numerous to mention, Rich is also really heavily involved in education, both at the graduate school and medical school level. He's also taught quite a bit in the neural systems and behavior course at Woods Hole, and we're just phenomenally lucky to have him here. So I won't say any more, but here's Rich Mooney.
Thanks very much, Sam. You can all hear me all right? Great. It's really nice to be here. Real pleasure to be back in Atlanta and to visit all of you at Emory. It's a really interesting concentration of computational neuroscience approaches, which I think is a great harbinger of good things to come. Okay, so I would like to talk to you about research coming out of our lab over the last few years. I'm going to review some published data, and I'd like to also get into some recent experiments that are unpublished that I think are really going to be exciting. So I want to make sure I get to that, but I also want to make sure that I invite you to interrupt me if things are unclear or if you have questions. So please don't wait till the end. What I want to talk to you about today, and really the take home, is that early sensory experience is important to behavioral learning, directly imprints and alters motor circuits that control behavior. And this is in contrast to the traditional view that sensory experience first transforms sensory networks in the brain and then only more slowly over time reshapes the motor circuits that control the behavior that comes from that sensation. So, by way of introduction, and before I talk about birdsong, I want to just introduce a classical modular view of how the brain works, stemming from 19th century neurology from Broca and Wernicke, respectively, that showed in human speech production and comprehension that there was some localization of these processes to the frontal and temporal parietal regions of the brain, respectively. And this is certainly the case. People with lesions in Broca's area have a major problem in producing fluent speech, whereas people with lesions in this region, the tertiary auditory cortical region that Wernicke described, have primary deficit in speech comprehension. However, the reality is that these structures, if you look at, as I'll show you in a moment, if you look at this in more detail, that there's really mixed involvement of comprehension and production with lesions in each of these areas. And a classical view of how experience affects the brain is in the visual cortex with monocular deprivation, evidence that systemic deprivation of peripheral input by monocular occlusion can lead to expansion of the non-occluded eye. The representation of the visual cortex expands and shown in these autoradiographs where the whiter regions represent input from the non-deprived eye. So it's clear that early sensory experience matters. And the question is how early sensory experience, particularly early auditory experience, is important to something akin to speech learning, song learning in birds, affects a brain that's organized in this modular manner. And again, as I mentioned at the outset, the take-home is that motor regions involved in producing, in this case, birdsong, are immediately affected by early auditory experiences critical to learning. And my guess is this is probably true in the human brain as well, in part because when we look at the connectivity between these modules for speech, comprehension, and production, they're massively interconnected. And given those interconnections, it's almost inevitable that early sensory experience acting through auditory cortical region is also going to impact the frontal regions that they speak. That's the big 30,000-foot view of the problem. What I'm going to talk to you about today is song learning in birds, which may seem a little more esoteric, but it has some really great power for explanatory power for studying these kinds of problems. And zebra finches like these shown here, including this juvenile in the foreground and this adult male in the background, engage in a process of cultural transmission of vocal behavior, much like speech dialects are transmitted from one generation to the next in our own cultures. And this is a two-stage process. There's an early auditory memorization of the tutor song. The tutor sings, the pupil listens, it forms a memory in the pupil's brain, and then more slowly there is a period of vocal rehearsal where the young bird uses its own vocal apparatus to sing. It compares its own vocal efforts through auditory feedback to this memory, and somehow this information is magically compared to the brain. And so there are really important questions here. So the phenomenology is that this memorization process can be really fast. It can occur within a few minutes of total auditory experience. Maybe a few hundred renditions of a second-long song from the tutor are sufficient to engage the learning process faithfully. And this process of motor learning is really, really slow. It takes weeks or months, and it can take hundreds of thousands of renditions of the bird's own song to refine it, to make it a good match and transform it from this inchoate song recorded early in the singing stage of this juvenile to a very good copy of the tutor song. And again, these are sonograms, frequency versus time plots, where this is about 0 to 10 kilohertz, and this is about a second. So this is really, really fast, and it's late in time relative to the motor learning process. And again, that supports the idea that maybe this auditory learning that happens early doesn't really need to involve motor areas at all. In fact, if you looked at it just from a behavioral perspective, you'd say it doesn't because the behavior doesn't change. I'll just play a little video of this so you can get a sense of how dynamic this interaction is. I joke that this is a little bit like some human dialogue that's fairly one-sided, and the one who's doing all the listening just falls asleep when it's over. But this has a lifelong effect on the pupil nonetheless, even though he was trying to appear disinterested. And so really the question is, where is this auditory experience stored in the brain? What I'll tell you about today to get at this problem is the background about neural circuits for singing, something about auditory pathways in the bird's brain, a more reductionist view of neuron circuits and synapses, and then recent experiments we've done using 2-photon imaging methods to explore structurally, literally at the level of single synapses, how the brain structure is affected in premotor regions by hearing a tutor song. And the take home there is that when the pupil hears a tutor song for the first time, dendritic spines on these premotor neurons are rapidly strengthened and stabilized. And then I'll show you experiments that we've performed that try to move beyond these correlative measures to establish that these structural and ultimately functional changes are part of the memory encoding process. And then at the very end, I'll talk about evidence that this same part of the circuit handles auditory feedback information. And so again, if you put this back into the behavioral process of song learning, the bird has to memorize the song and then use auditory feedback to evaluate its performance relative to this memory. And what I'll tell you is that this part of the brain that we're going to look at in a moment has some of the features necessary to do this kind of evaluation. Okay, so something about the bird's brain. This is a sagittal view of the bird's brain. This is the telencephalon, the thalamus, the brainstem, cerebellum. And for today's talk, I'm going to concentrate almost entirely on this nucleus HPC. And if you were a songbird neurologist, if you were like a broca of the bird song world, and you had a bird that came in with a lesion in HPC, you'd say, I've discovered a bird with a Broca's aphasia, because what it can't do is it can't sing. It can actually make a wide range of agonistic vocalizations, alarm calls, contact calls, and the like perfectly well, but it can't produce this learned song. And that's just like the broken aphasia in humans, where they can make emotional vocalizations, cry, moan, laugh, and so on normally, but they can't speak. And HVC has an output cell type that projects to this motor nucleus RA, which in turn projects to the vocal respiratory plant in the brainstem that controls phonation during singing. And HVC, so that could explain how HVC lesions affect learned song. And HVC has another output cell type that projects into a basal ganglia homologue in the bird's anterior part of the forebrain, and we call it split X. And this part of the pathway shown in red here isn't necessary for the bird to produce its adult song, but it is necessary for the bird to learn properly. So if this pathway is damaged, then the bird doesn't learn normally. In fact, it can't copy if the output of this pathway is lesion. Now, again, for nomenclature, I'm going to refer to HVC-RA cells and HVC-X cells to distinguish these two different output cell types. But you can think of these as, if you're in the mammalian literature, as the equivalent of neuron that's projecting, say, from premotor cortex to motor cortex and then onto the motor neurons. And this is a corticostriatal population of cells. Now, again, as I mentioned, if you lesion HVC, they can't sing. If you cool down HVC, the bird's song tempo dilates, it becomes slower. If you electrically stimulate an HVC while the bird is singing, it resets the song to the beginning of the song phrase once the stimulation is stopped. These are all kinds of evidence that point to a motoric role for HVC in singing. And in fact, when one records single unit activity from these HVC-RA neurons, these cells that project to the motor nucleus RA, during singing, and recorded the zebrafinch that produces these polysyllabic motifs or phrases 3 times in a row, a single HVC-RA neuron bursts once per motif, very precisely, very, very high frequency, about 400 or 500 hertz, for about 10 milliseconds. And if one records from different neurons, the HVC-RA neurons, in a single bird and aligns them to the parent motif, they form a time series. So this has lent itself to the idea that there's a kind of a clock for singing, kind of a tempo apparatus, and we can come back to that towards the end because I think some of the implications of the experiments I'll show you are relevant to that. All right. Now, something that isn't shown in that previous diagram is that there are also auditory inputs into this structure, into this pathway, or these pathways, and that's maybe not to be unexpected given the dependence of singing on auditory experience. But the place where as far as we've been able to tell where this information enters the network is through HVC. So this makes HVC the sensory motor nexus. This is the place where auditory and premotor signals are aligned. So the first set of experiments we were interested in asking is, could we even begin to study how HVC changes during early auditory experience? And so the idea was, could we go into, kind of in this kind of fanciful painting on the right, could we go in and look with a really good magnifying glass into the pupil's brain right around the time the tutor was singing and get images like these of dendrites and spines and use this as a way of evaluating information about connectivity and the structure of the neuropil in the bird's brain. Again, for non-bird experts, so birds are just like mammals in that their central neurons use glutamate and used as a primary excitatory transmitter, and they use a mixture of AMP and NMDA receptors located primarily on spine heads, just like cortical neurons in the mammal. So at this micro level, it's very hard to tell these synapses and neurons apart from their mammalian counterparts. We're not dealing with something very exotic at this level. It's a kind of a generic vertebrate ground plan for building neural circuits. But again, just as in the mammalian brain, the dendritic spines, these little protuberances here coming off the dendritic shaft, are the major sites of excitatory synaptic contact. And people use these as a proxy for measuring synaptic connections and strength. Okay, so how you do this is take the bird, inject it bilaterally in HVC with a lentivirus construct that encodes for the green fluorescent protein, GFP, and wait a couple of weeks for it to express at high levels in infected neurons, and then build a little cranial window over HVC. And this is a really convenient aspect of this system is that the most important structure in the bird's brain just happens to be put right on the dorsal surface where we can visualize it. That's kind of a joke. What I mean is that we're just taking a little keyhole to look into the bird's brain, and it happens to be that HVC is one thing we can get to easily, and that was just blind luck, but it turns out to be a great advantage here because with a little cranial window mounted on the bird's skull, we can peer with a high-powered projector into the bird's brain and image these neurons like this striatal projecting neuron in HVC. And when we do this, an image over short time windows, I should say that these experiments are done in this manner. The bird's injected about 2 weeks before the first imaging session starts with the virus. We wait for high expression. We window the bird. We begin to image during the bird's subjective nighttime. So the birds are maintained on a reverse day-night cycle, so we don't have to stay up all night doing the imaging experiment. But the consequence of this is that the bird is allowed during the daytime to sing, to associate with a tutor if we want it to, and so on. So the first observation just is that over short time windows, most of the spines on a segment of dendrite are stable. These are marked by S's. But some spines emerge and others disappear. And in other systems, these have been shown to correlate with the emergence and disappearance of excitatory synapses on the neuron. And I'll show you some electrophysiological evidence suggesting that the spine changes we see are actually reflecting changes in excitatory synaptic transmission. So you can take the gain and loss of spines and calculate an average of turnover rate, and you can measure this throughout development. And I didn't emphasize this, but in zebrafinch development, sensory learning from the tutor ends about 2 months after hatching, and sensory motor learning starts at about 45 days or so and extends up to about 90 days. So these are, in this species, normally overlapping. And if we just image HVC neurons, these are now projection neurons of both types, those are spinous neurons, and image over the course of development all the way from 45 days up to 90 days, absolutely nothing happens. This was the biggest negative result involving a lot of work that we ever did in my lab, and I actually take great credit for this because I was able to inspire my postdoc Todd Roberts and my grad student Katie Cheetah to do these control experiments. And we thought when we first did these, we thought, oh, well, you know, this is a motor structure and the bird's undergoing a lot of behavioral change. This structure is driving the behavior. We'll see a lot of change in spines, but in fact, it's perfectly flat, despite the fact that the song model is not even close to being reproduced here, and then it's being produced almost perfectly here. So those are all birds that have been raised from the time that they hatch from the egg with their tutor, so they've been having normal auditory experience. And what got interesting is when we raised birds as isolates, so they're raised without a tutor, and we measured turnover, it was really elevated at this early stage. And if we went out to late stages in development, out here, when these isolate birds had stabilized their own very simple isolate song, so even though they don't copy from the tutor, they're committed to singing this very impoverished song that females don't like very much, and they're kind of stuck. So learning actually is beneficial, I should also stress. You know, it matters what you sing, and birds that don't learn anything get ignored by the young females who want to mate. But the really interesting part came up here, right at this time point. And when we looked there, some of the birds had elevated turnover like their younger counterparts, and some had low turnover like their older counterparts. And what we knew from other behavioral experiments is that this is the roll-off period for memorizing tutor song. Even in isolates, they have to give up at some point. The brain becomes refractory to learning from the model. And we thought, well, that's interesting. So now we have birds, these are means for individuals now. We have some that are elevated and some that aren't. And we know that younger birds that are raised in isolates up to this point will copy from the tutor no matter what. We thought, okay, well, maybe this is telling us something about the commitment of the brain to an isolate lifestyle. And we thought if that's the case, we can take these birds after we've imaged them, and we can tutor them for a few days, then raise them up to adulthood and assess their learning outcomes. And 10 to 15 years ago, this is an experiment you couldn't do because to get this measurement of spine structure out of the bird's brain, you'd have to kill it. But with 2-photon imaging, we could actually get the information out and then just put the bird back in with this tutor and ask what happens. And in fact, when you raise birds up to adulthood, in fact, just those birds I showed you, the learning outcome varies. With some birds copying a fair amount from the tutor and some not at all. And you can then take that information, and align it with the amount of turnover measured before the tutor was introduced, and there's a correlation. I should point out that we're measuring typically 1 neuron per bird. There are about 100,000 HVC neurons, and we're just kind of infecting them at random, so kind of at low levels. So low incidence. So even though we're making measurements just from a very small fraction, maybe 1,000th of a percent of the available neurons, we're still seeing a signal that correlates with a learning outcome that's going to take a couple of months to evolve. Okay, so that's one thing. So the take home is that if you have higher turnover levels, it looks like you're more likely to learn from the tutor if he's introduced in time. So the other thing is we could take some of these birds and image them across the tutoring divide. And this was just, again, these were early days when we were getting the hang of this technique. We could get some animals imaged repeatedly over many days, sometimes not. But again, luck would favor us, and we got some birds from the low end of the distribution and some birds from the high end of the distribution. So these birds marked in red are high turnover learners. These birds are low turnover dummies. These ones would get the little bumper sticker to go on their car. They're honor students, and these ones I don't get a bumper sticker. Okay, so it turns out that if you look at changes in turnover from the night before the tutor was introduced to the night after the tutor was introduced for the first time, the low turnover group doesn't change. Turnover is pinned at a floor level. In fact, this is very similar to adult levels. Whereas the high turnover group that will eventually learn shows a 50% reduction in turnover. So that's night to night. That's the highest temporal resolution we've been able to get so far doing this kind of experiment. But within one day and one night of experience, the brain has been transformed at this level. And it's been transformed in that the dynamics of spines has been reduced. Now, you may notice something about this, namely that even in the case of the high turnover group, if you take this value and subtract it from 100, the vast majority of spines are stable. So we're looking at actually a very small fraction of spines in the nucleus. And it turns out that the stable spines get quite a bit bigger, they get brighter, and I'll show you some data on that as well. Now, the question is, does this have a functional correlate? So all I've shown you right now is structure, but the function actually is revealed in these current lab recordings. These are now made the night before tutoring and the night after tutoring, just like the imaging experiments, but now instead of imaging, we're using a sharp microelectrode to poke different projection neurons in HPC. Now, this lacks the beauty of the two-photon technique, where we can actually go back and interrogate the same cell from one night to the next. But we're recording in the same small patch of HPC. And what you can see by eye is that there's an enhancement of the spontaneous synaptic activity. These are, again, for non-electrophysiological types in the audience, these are reflections in voltage. So these reflect spontaneous synaptic events on the cell. And what you can see is the emergence of this pronounced compound bursting behavior, as well as an elevation in the more isolated events. And if you just take a cumulative distribution of these spontaneous depolarizing postsynaptic potentials, there's a very strong rightward shift in these isolate birds the night after they socialize with the tutor. Okay, so that says that there's this big, physiological effect, and I'll come back to this a little bit later. This compound bursting tells us something important. We've spent a lot of time recording the HPC, and we know what drives that compound bursting. It's a major auditory input into this nucleus, a structure called Neurobiology. So we think what's happening is that synapses from this auditory region are being potentiated and strengthened by hearing a tutor song for the first time. So I mentioned many of the spines are stable, and what you can see is that stable spines in these high turnover birds go from being certain brightness to being much brighter. From the night before tutoring to the night after tutoring. And the brightness is simply a reflection of the number of GFP molecules that we're imaging in the field. And those number of GFP molecules correlate with the volume that they're filling, in this case, the spine head. And other studies have shown that as the spines get brighter, the spine head is getting bigger, the postsynaptic density is getting bigger, and the number of AMPA receptors incorporated in the spine head is increasing. So again, this is a structural correlate of synaptic strengthening that, along with the electrophysiological data, say that there's a big bang happening in this part of the brain, in this motor part of the brain, when the bird hears its tutor for the first time. Now again, these effects on stable spines are specific to these high turnover birds that will learn. And in fact, if you look at relative spine brightness, it's actually high in these isolates with low turnover. So they have more stable, bigger spines to begin with, whereas the high turnover animals have dimmer spines that get significantly brighter once they've heard the tutor sing. So that's just showing those data in a different form. But the take home is there's a massive change to synaptic input on these premotor neurons. So even though clinically you would say this is a motor area, this formative auditory experience, one day of this formative auditory experience, maybe hearing the tutor a few hundred times in total, is sufficient to transform the network. It's like the brain is waiting to get this experience, and when it does, it kind of galvanizes the whole circuitry. Yes?
I was wondering, do you know which subtype in terms of their projection?
Yes.
Do you see these?
This happens in both projection neuron types. Okay. So that's really nice. It's a correlation, and it's a really interesting correlation, but what we were particularly interested in doing is getting beyond the correlative. So I'm going to show you a few different types of experiments that we've done to get at this. As I mentioned, these are neurons that use glutamatergic transmission, and they have a mixture of AMP and NMDA receptors. Spine enlargement like this, synaptic strengthening, is thought to depend on NMDA receptor activation. So we took an educated guess that that's what might underlie increases in spine size, stable spine size in HVC. And to test this idea, we took isolate birds, we introduced the NMDA receptor antagonist into HVC and imaged these HVC neurons before and after tutoring. And again, these are data from birds that have either been in this low turnover group or high turnover group without the NMDA receptor blocker. These are birds that are high turnover birds that have been treated with the NMDA receptor blocker. So spines fail to get bigger when an NMDA receptor blocker is present in the HVC, despite the experience of the tutor. And we can now take this to a behavioral level by putting microdialysis probes into HVC bilaterally and essentially doing the same experiment but reading it out to adulthood. So the design is the birds are isolated till this 5-day window where they spend each morning with a tutor. And experimental animals have the NMDA receptor blocker on board while the tutor is present. Their siblings are implanted like this. They have saline on board while the tutor is present. And then there's a reverse. So the birds that are treated with the blocker in the morning get saline in the afternoon, and the control birds get the NMDA receptor blocker in the afternoon as the tutor's after the tutor's been removed. You raise the birds up to adulthood and assess learning, and all the APV-treated birds fail to copy. So that's just at the same chance. An isolate is scored this way. Okay, we're using this similarity metric to the tutor song that I'm showing here. Whereas the control birds actually learn normally, which is surprising to us because they're still actually practicing. We would expect, well, maybe there's some important consolidating step that depends on NMDA receptor activity after the tutor's been removed. And in fact, this says you can block NMDA receptors after the tutor's been removed, just an hour or so later, and learning proceeds normally. So that narrows the time window quite a bit. And if you look at the behavior, again, this may be more informative to the birdsong experts in the group, but if you take the tutor song sonogram and you look at the adult songs of these APV-treated birds, they have features that are actually completely characteristic of isolates. The syllable durations are much longer. You can find frequency upsweeps that are unusual and so on. And you can see that it lacks this really beautiful structure of the tutor song.
How long did they give you to watch that?
Probably a couple of hours, yeah. Hour or two. Okay. All right, so that's pretty good. But the limitation is that we still have a situation where the tutor is present with the pupil. The NMDA receptor blocker is on board, and as Sam was just alluding to, it's sticky, it sort of stays around. So it's blocking NMDA receptors while the tutor is singing to the pupil, and it's also blocking NMDA receptors while the pupil is singing in the tutor's presence. So again, maybe during the couple of hours that they're co-housing, the singing that the pupil does is really critical to burning in this memory. You need NMDA receptor activity in HVC for a motor-related function that doesn't have to do with listening to the tutor song. So we used a different approach, which is to use viruses to express channel rhodopsin. This is a light-activated cation channel. It's in wide use, maybe too wide use in the neuroscience community. This has got a GFP tag on it, so we can check the distribution of infected neurons. This is using a self-complementary AAV, so we can get very fast ramp-up of channel rhodopsin expression. And this is schematic, and this is a picture of the labeling. The really critical feature about this is that the infection can be restricted spatially quite well to HVC, and we're going to put a light pipe in over HVC, and we can make electrophysiological measurements to confirm that it doesn't extend even into the ventralmost aspect of the nucleus, let alone outside of the nucleus. So this gives us a spatial restriction that's quite good. We can go in a pupil or a potential pupil bird, and we can use light to evoke multi-unit activity to confirm that we've got good functional expression. And I wanted to say something about this expression, and this functional activity. Here's the light bar, and these are different light pulses from different recording sites in a single bird. And what you can see is that these are rastergrams, these are individual trials of light, and these are spikes represented by dots. And what you can see is that some sites show fairly faithful following of the light. Some lag the light considerably. Some show suppression followed by excitation. This is a network level disruption. So we're infecting both the projection neuron types, which are excitatory, as well as local interneurons, which I haven't talked about, which are inhibitory. And that, a result of that is when they shine light on, you get complex effects. But I just draw a point here that even though the temporal fidelity is pretty good, the scale that we're going to work on is even too fast for channel rhodopsin. So I'll address that in a minute. And we're disrupting things at a network level. Okay, so the experimental design, and this is something that is, I think, the first time optogenetics have been used this way. So usually the experimenter sits there and flashes light on the mouse's brain or whatever it is, and the mouse turns in the direction, for example. But this is something where the tutor is going to gate the laser. So what the design is that we use software to detect when the tutor is singing, and we can actually use this software and tune it so that it selectively recognizes when the tutor is singing versus the pupil. And it distinguishes songs from calls and occasional songs. So the tutor is going to sing, and when he sings, he's going to gate the laser that is connected to these fiber optics to the pupil's HVC.